We're delighted to bring you the seminar presented by Labroots. Welcome everyone, and thank you for joining us for today's keynote presentation titled The BRAIN Initiative and its Promise for the Treatment of Neuro, Mental, and Substance Abuse Disorders. It's my pleasure to introduce to you Dr. Walter Koroshetz. Dr. Koroshetz serves as the Director for the National Institute of Neurological Disorders. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions should arise during this presentation, we encourage you to submit them in the Q&A box. Our speaker will respond back to you via email. And as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Koroshetz. I will now turn the presentation over to him.
Hi, I'm Walter Koroshetz. I'm the Director of the National Institute of Neurological Disorders and Stroke here at NIH. And it's a pleasure to talk to you today about what I think is the most exciting neuroscience project ever, we call the BRAIN Initiative. And just to give you a little bit of background and what our objectives are, the plan today is to help you to understand how the BRAIN Initiative and the new technologies that are being developed in this initiative will enable really powerful means to monitor, modulate, and map brain circuits. So the 3 M's: monitor, modulate, and map. And the focus is on brain circuits. Also, because we are the National Institutes of Health and the Institute for Neurological Disorders, we are really invested in the BRAIN Initiative because we think it will bring promise for these new technologies to more effectively diagnose and treat neurological, mental health, and substance abuse disorders. Some of these technologies are going to be powerful in the medical community, but because they involve monitoring, modulating brain circuits, they probably will have potential to move out of the medical space. And even in the medical space, but more so outside of it, there are going to be some serious ethical questions that society will have to address to understand what is the appropriate use of these new technologies. So we're going to talk about and end up with a talk about neuroethics. Just to go back, the National Institutes of Health is the taxpayers' investment in biomedical research. And the NIH's mission is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce illness and disability. The NINDS mission is really a mirror image of this, the focus being to understand how the nervous system works and to use that knowledge to decrease the burden of illness due to neurological disorders and stroke. Of note, the taxpayer has been incredibly generous to the biomedical research infrastructure here at NIH, and the budget, which was flat for years, 12 years until 2016, has seen increases in the last 4 years and another one projected for 2020. And so the budget of NIH is $39 billion a year. And 80% of that money goes to over 50,000 competitive grants, 300,000 researchers at more than 2,500 institutions. And there's a mandate for about 3.5%, 3.6% of that money to go to small business grants to develop products that can improve health based on the knowledge coming out of NIH-funded research. Now, it's a very feudal system. There are 27 different institutes. NINDS, the Neurological Institute, is one of them. 24 of these institutes and centers actually award grants. So depending on what your area of science is, you may be more involved with one institute as compared to another. And many of these institutes, which we'll get to, fund neuroscience. So actually, the neuroscience budget is about $9.47 billion. So almost a quarter, a little less than a quarter of the NINDS budget goes to neuroscience. And that budget is split among about 13 or 14 different institutes. And important to note is that if you look at all the areas of science, neuroscience is actually the one that receives the largest amount of funding, more so than cancer, more so than heart disease. Now, the purpose of the BRAIN Initiative is to revolutionize our understanding of how the human brain works. And the impetus is to try at the end of this to make a difference for brain disorders, which are the leading source of burden of illness in the United States. So if you combine mental health disorders, neurological disorders, and substance abuse disorders, they are the leading cause of disability in the United States. And the BRAIN Initiative is trying to figure out how we can understand how the brain works so that we can better diagnose and treat those disorders as its eventual goal. So I want to just simplify the problem for you for just a moment and ask you to think of, in broad terms, about the fundamentals, workings of the nervous system. What do we know and what do we need to know? So I'd argue that, very simplistically, what we do know is, as Galvani discovered, neurons transmit electrical pulses along their processes. And Hodgkin and Huxley discovered how these action potentials are propagated along an axon. Now, the way the neurons communicate with each other through neurotransmitters discovered by cats, we know that signals can be transduced from the end of the electrical stimulus at the axon to the postsynaptic neuron through these chemical neurotransmitters. So those are the kind of the fundamental processes that we understand. Now, Cajal was the first to see how these neural cells are connected into dense networks. And its information processing is going on in these dense networks. But that has been the problem. How do you see the firing patterns of millions of interconnected neurons in activity and similar numbers of glial cells that give rise to a behavior, a memory, a perception, a thought, a series of thoughts? So the computer analogy is, how does the brain process information like a computer? How does a computer process information? What are the computations that are being performed at each of the levels inside the brain? The communication analogy is, if action potentials can be seen as the letters in the communication strategy, how are these letters formed into words, the words into phrases, the phrases into sentences, sentences into paragraphs, paragraphs into chapters, and these into the books that represent our mental life. So this knowledge of how the networks of neurons process information to allow our mental life is really the area that has been very hard to crack. And we don't understand the fundamental principles that underlie information processing in the brain. And that's what the focus is of the BRAIN Initiative. But to understand and get to this kind of information, we need new tools. And we're taken to a quote from Freeman Dyson, who was a philosopher and mathematician at Harvard, who said that new directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained. And from what we talked about in the last slide, what we need is a tool-driven revolution to discover how network activity in the brain leads to our mental life, our many behaviors, thoughts, language, and so forth. Now, technology has had a tremendous effect on understanding the nervous system, particularly if you think about in my career coming out of medical school in the mid-70s when there was the first CT scan. We were quite excited to see a living brain in a person using X-ray tomography. But look at that picture from 1974. One really only gets the resolution to be able to see the size of the fluid-filled ventricles, those black areas in the middle. Very little about the structure of the brain. But over the next decades, MRI came on the scene, and the type of resolution that you can get with an MRI scan is almost at the level of what you would see if you cut a brain and stained it, seen here on the right panel. And in fact, MRI has completely revolutionized the practice of neurology, neurosurgery, where there are structural lesions that one is interested in and following and detecting. And yet it's had very little impact in areas such as psychiatry, substance abuse disorders, where there are not major structural changes, but the circuit function can sometimes be extremely disordered and yet oblivious to our ability to see. Now, can one think about trying to see that type of information in the brain? And that's what the BRAIN Initiative is about. And the areas that I want to point out are the complexity of the problem, but also the imagination of scientists to allow us to kind of crack into this code of information processing. So the video that's going to show next is just an example of the complexity of the interconnections between neuron areas in a mouse brain, a slice of mouse brain. And the technology, the really earth-shattering technology, is a technology called Clarity, in which scientists called Deseroff at Boyden develop a technology to take the lipid out of the brain without affecting the structure. And that makes the brain transparent, translucent, so that if you have a fluorescent marker for any molecule you want, now you can see that how that molecule is arranged. And here you'll see a molecule that stains neurons and their processes. So please play the video. The video that you just saw has complexity in it at one scale, but as we'll show later, the problem with understanding circuits of the brain is the immense difference in spatial and temporal scales that have to be understood. And we'll get to that in a second. But again, to emphasize, the BRAIN Initiative is trying to get to see circuits in action, to understand how the brain controls, how we move, plan, execute actions, and remember, how to monitor and manipulate circuits for improved neurological or mental health function, and how disordered brain circuits cause these neural, mental, or substance abuse disorders. And as Freeman Dyson wrote, to try and get at this problem, we need new tools, we need new technologies. And so the BRAIN Initiative has a focus on technology development, and actually the N in BRAIN stands for neurotechnology. So BRAIN Initiative is brain research to advancing innovative neurotechnologies. And the first 5 years of the BRAIN Initiative, which we are just now ending, had a tremendous emphasis on technology development and led to abilities that no one really imagined we'd be able to get to in 5 years. The second 5 years of the BRAIN Initiative, which we're now embarking on, has a heavy focus on using those technologies to do discovery-driven science and increase our fund of knowledge about how the brain works. But the second 5 years will continue to have a similar focus on neurotechnology development and dissemination as well. The BRAIN Initiative has really 8 pillars, and I'm going to go through these so you can see what it looks like in totality, and then we'll be giving you certain examples, but very small number of examples in each of these areas. The first pillar is understanding diversity of cell types in the brain. So the human brain has, by best estimates, 85 billion neurons, a similar number of glial cells. And so the diversity of the brain cell types is tremendous. And we don't have, at this point, a very good understanding of that diversity. So the first pillar of the BRAIN Initiative is to try and understand all the different cell types in the brain. Now, as we talked about before, and as Cahal indicated for the first time, these cells are not sitting isolated in a dish, but they're interconnected, and they're heavily interconnected. And so what we'd like to do is to understand those connections. And the second pillar is then to map the circuits of the brain, but at the multiple scales that are important. And this there is the problem, because information processing is occurring at the molecular level, say at the synapse level, or at the ion channel level. So nanometers scales to information processing across synaptic junctions to a cell with multiple dendrites, multiple axons, and then these cells are interconnected into a nucleus. The nuclei are interconnected across the brain. And so the spatial scales at which information processing is occurring is really quite daunting. And so the maps are occurring at different scales. The ultimate, however, is to see circuit activity in action and as the brain is driving behaviors or processing information. And that requires the ability to see brain activity, either the firing of neurons, the calcium changes that occur when neurons firing, or neurotransmitters crossing and activating postsynaptic cells. If one could see all the brain cells and their activity with regard to a behavior, then one could develop potential models that correlate neural activity to a behavior. But those would be correlations. To try to get from correlation to causality, what needs to do is to build the model and then change the activity of the neurons and see if the model then predicts the change in the behavior. And that requires modulating the neural activity. And so technologies that would allow one to do that, to understand causality in terms of how neural network activity leads to a behavior, that would be a tremendous goal to attain. Now, when we first showed the slide with what we know on one side and what we don't know on the other side, we talked on the side we know about really fundamental principles, how the cells are connected, how information is processed from one neuron to another. But in the area of understanding information processing, we have no understanding of the fundamental principles or the processes that are the computational processes that go on to lead to a behavior. So understanding these fundamental principles, what I call the language of the brain, the words, the phrases, the sentences, these are the type of computational side of the BRAIN Initiative that's going to be very important. And as one might imagine, many of the work that we talk about is best done in animal research, but we really would like to understand how that research in animals translates to understanding of the human brain, and then to go from understanding these pieces, the maps, the brain in action, causality, to give a more holistic view of brain function. So that's the seventh pillar, from BRAIN Initiative to the brain. Brought out in the last advisory council, the director on the BRAIN Initiative was another pillar called the organization of science. So neuroscience has been predominantly done in single laboratories, single PIs, communicating, collaborating on particular projects, but to understand how the brain works and to really try to understand how all this information is processed across tremendous temporal and spatial scales, we require a real team effort. And so in the BRAIN Initiative, there is an emphasis on building teams to solve big problems that are really hard to get at, particularly in terms of scaling up our knowledge into how the brain works. Now, we're very fortunate that the BRAIN Initiative is funded by Congress to the level now that was in the professional judgment of the advisors that put together the first BRAIN 2025 report. That was kind of a roadmap for how the BRAIN Initiative should go ahead. They recommended that a budget somewhere around $500 million a year would be what would be needed, and that's exactly where we are now. So we're really grateful to the Congress for understanding the importance of the BRAIN Initiative and supporting it. The BRAIN Initiative in its first 5 years has led to a tremendous number of new technologies and new knowledge about how the nervous system works. There have been over 900 publications on, say, understanding how to image thousands of neurons in an awake behaving animal, or developing super resolution imaging of molecules using particular signaling molecules that will blink, and the characteristics of the molecule that's being stained can be encoded in the actual blinking kinetics of the imaging marker, or ways of doing stimulation using light of neurons, so that one can actually stimulate multiple neurons at the same time using optogenetic techniques that will cause the neurons to fire in a pattern that's up to the investigator to set. And this has led certainly to some of the ethical issues as well that we'll talk about in the future, next couple of minutes. And the BRAIN Initiative, to say simply put, is actually also funding research in neuroethics related to the BRAIN Initiative. So if we think about it, is understanding how the brain knowledge into understanding the human brain and treat disorders of the nervous system. Now recall that the first pillar of the BRAIN Initiative was understanding cell diversity. 85 billion neurons in the human brain, 8.5 billion in the mouse. And this has actually been the signature project of the BRAIN Initiative so far. And that's because high throughput methods became available to do single cell analysis of brain tissue. And this has allowed scientists to identify a transcriptomic signature for different cell types. But instead of doing it at 100 or 200 cells at a time, it can now be done at up to 1 million cells at a time. And this has launched a team of scientists to do the brain cell type atlas of the mouse. And this is well on its way. The mouse has 8.5 billion neurons, and the transcriptomic analysis will give us a scaffold that identifies different cell types based on their transcriptome. But of course, there are other characteristics of the cell, the lineage, the connectivity, the firing characteristics, the morphology, that all need to be filled in as well and added to the transcriptomic cell characterization. But those latter areas are much lower throughput, and the ability to really get at the entire mouse brain really rests now with the high throughput abilities to do single cell transcriptomic analysis. I should say that an incredibly powerful consequence of the Cell Census Project is the development of tools that give you genomic access to particular cell types in a very precise manner. And that's because the transcriptomic analysis is giving folks the ability to identify cell type specific enhancers or transcription factors to which genes can be hooked up. And these genes, say a gene that leads to the expression of a channel rhodopsin, allows you then with, say, light to turn on or turn off a particular cell type. And so with the transcriptomic knowledge, we're now able to have a cell type specific genomic key into these different cells. This is actually a very big brain team project, Brain Initiative team project, and much of this is being led by the Allen Institute, which is an institute that's dedicated to understanding brain circuit activity. And so this was incredibly fortunate to have this group assembled and ready to take on this tremendous project. The next video I'm going to show you is an example of some of the technologies that have been developed to be actually see with light microscopy across large brain regions very quickly. And the purpose of this is not only to be able to see structure, but also to be able to see, and I'll show you in a bit, the activity which is captured in optical signals as brain cells fire. So this is from the Hillman Lab at Columbia, very, very high speed volumetric microscopy. Please show the video. Now I want to tell a little story, which is really, again, a fascinating one, something that one would not have predicted, but has indeed had significant impact. So when we are trying to look at brain circuit function across the brain, the scales range from nanometers to, multiple centimeters. And if one wants to get really intricate detail, one would have to do electron microscopy throughout the whole brain. And certainly people were doing this, and we'll show you some of that. But that is incredibly labor intensive, time intensive, and expensive. and not very translatable to multiple different laboratories. I did talk to you previously about the technique called clarity, which allows you to take the lipid out of the brain, make it transparent so that you can label with an antibody, fluorescent antibody for a molecule of your interest. And you can then see how that molecule is distributed in the brain. Very powerful technique, but it's at the light microscope level. How would one, say, get to the resolution where you could see connected to a molecule on the other side of the synapse? Right now, you would need electron microscopy. Interestingly, Ed Boyden at MIT developed a technology to get you there with light microscopy. And what he did was to use a type of clarity method to make the brain translucent. And then he embedded the brain in a polymer and then added water, and the brain expanded. But the structure was not distorted. And with this expansion technology and then using a highly developed light sheet microscopy, he's able to see and scan across an entire brain, but can actually see molecules such as the pre- and postsynaptic molecules, because now with the separation that occurs with the expansion technique, those molecules are separated from each other enough to be seen with light microscopy. So I'm going to now show the video. This video was an entire Drosophila brain, and they have been able to do this within about 3 days of work, of scanning, an entire mouse cortex also at the same amount of time. And of course, with electron microscopy, that would take many months, maybe even a year. So interesting technologies that one would not have predicted, but have shown to be really incredibly important. Now, the next video, which I'm going to show, is from Jeff Lichtman's lab, Bobby Castori, and it's a very tiny, tiny piece, millimeter piece of cortex. But what it does show is the complexity at the very fine temporal scale. So we've seen, in the first video I showed the mesoscale, and this is the real microscale. Please show the video. So this video demonstrates the complexity of the connections between not only the neurons, but between the glia and the neurons. And to get a complete knowledge of how brain circuits are functioning, we really have to understand, from this level all the way up to across the brain level. Now, the next pillar, besides maps, is how do you monitor brain activity? For years, scientists would stick an electrode in a neuron in a behaving animal, record electrical activity as the animal sees particular stimulus or makes a certain movement, and try to develop a model for how that activity led to or was involved in the behavior. But I'd argue that that's similar to trying to understand Gone with the Wind, and you can see the movie over and over again, but each time you can only see one pixel on the screen. Analogy to what we past have been able to do with the single electrode recordings. Optical techniques now, where you can develop a transgenic mouse or into an area brain that will cause expression of a molecule that lights up when calcium flows into the cell, which will occur with action potentials, allows you to see, as you can see here, the activity of neuron firing in here, hundreds of neurons at a time. And the technology is now such that over long periods of time, when you substitute the opaque skull with, say, a plastic translucent skull. So now scientists have the ability, at least at the surface level, to be able to see a large number of neurons firing in a particular behavior. And the trick then becomes in how do you analyze this type of activity to understand the actual properties of the activity of the network that leads to a behavior. There are also genes that have been into neurons that will give an optical signal when a neurotransmitter, this one dopamine, binds to its receptor. So these have sometimes been called sniffers, and they have optical signals that allow you to see, at least in real time, of neurotransmitters. And this technology is just beginning, but many chemical essentially be labeled, and so one would have the ability to see chemical release all over the nervous system in a very specific manner. Another technology to monitor brain activity. Now, modulating brain activity and turning on or turning off neurons is also an incredibly powerful technology that's been developed and utilized in the BRAIN Initiative. The earliest technologies were with optogenetics, where a gene is inserted and with light, that gene will turn on a channel that will either turn on the cell or turn off the cell from firing. And you can do this in a very cell-specific manner and only turn on or turn off the nucleus of cells that you're most interested in. There are also people working on ways in which once that artificial gene is expressed in a neuron, that it can be activated by, say, an inert chemical that the animal eats, so-called chemogenetics, or by an ultrasound pulse from outside the head, so-called sonogenetics, or electromagnetic radiation from outside the head, called magnetogenetics. And each of these are working to some different degrees in animal experimentation at this time. So just to summarize, this allows one to have really precise ability to manipulate and modulate neural activity in a very precise manner using these new tools. This is a complicated slide, and I won't go through all of it, but it's just a very interesting example from Greg Shearer and Mark Schnitz's lab at Stanford. And so they're interested in pain. And so I take you to look at the bottom tracing, where you can see these kind of blue patches on the F. And on the left side, you see the experiment performed in a normal animal. And one side, called the test plate, is cold. And the other side, which is very dark blue, is a control temperature that the animal is perfectly comfortable with. And what you see is the animal spends all its time on the plate that it's at the temperature that they're very comfortable at. And then they show something very similar in an animal that's developed a neuropathic pain syndrome by a compression lesion to the sciatic nerve. Here again, you see the same behavior. If anything, the animal spends less time on the cold plate. And what they were able to do is to find cells, neurons in the basal amygdala that are responsive to pain. But the remarkable thing about these neurons is they use a technology, a chemogenetic technology, to turn off the cells. And when the cells are turned off, just these cells in the basal outer amygdala, the animals have normal ability to detect painful stimulus, but they lose all the negative emotion that goes along with the painful stimulus. So if you go back to F again, and you see in the neuropathic, on the right side, the neuropathic plus saline. So this is the neuropathic animal, and they give the animal saline. And again, you see it doesn't spend any time on the cold plate, all on the comfortable temperature plate. But if you give a chemical called CNO, or clozapine, it will activate an artificial receptor in the basal amygdala to turn off these cells. And then, although the animal can detect a painful stimulus, it doesn't really care. It'll spend as much time on the cold plate as on the comfortable temperature plate. So just an amazing discovery. If one was looking for a mechanism to treat chronic pain, this would be perfect. You don't want to take away someone's ability to detect pain, or they'll hurt themselves, but you would like them not to have that kind of negative emotional, painful perception, which is what causes their suffering. So an example of finding cells, turning them off for a really tremendous impact. Now, to get to where we want to go, we want to understand what are the grammatical rules for information processing in the brain. And this is going to take a tremendous push to bring people who are well-schooled in theory, modeling, mathematics, computational biology, to be able to see the data that the investigators are collecting, the network activity data, correlated with very precise definitions of behavior, and to try and understand what those correlations are, develop the models, and now with the ability to manipulate those networks, we can actually test the models, because if we have the right model and we make a perturbation in the firing pattern, we should be able to predict what the change in behavior is. So there is a tremendous amount and growing amount of research in this space in the BRAIN Initiative. Now I'm going to pivot a little bit to the potential and actual benefits to patients. So the BRAIN Initiative has neuroscientists, engineers, clinicians, and ethicists involved in understanding how the brain works, but there's also a group that is trying to jump the gun to bring this technologies to people to improve their burden of illness. And this is going on in research that is trying to manipulate circuits for health benefit in people with obsessive compulsive disorder, stroke, Parkinson's, epilepsy, chronic pain, depression, and even blindness. And we'll show some of those examples now. The first one I want to talk to you about is a closed loop deep brain stimulator that's been developed for patients with Parkinson's disease. Now, you may know that deep brain stimulation in, say, the subthalamic nucleus is a very effective therapy for the motor symptoms of Parkinson's, at least at the early and mid stages of the disease. There is, however, a potential side effect of the stimulation, and that is called hyperkinesias, where in Parkinson's one is usually very bradykinetic with slow, sometimes absent movements. But with overstimulation, one develops choreic movements, which are also annoying to the patient and to others. So how would one basically tune a stimulator to avoid the hyperkinesias? And that's what was done in this experiment. What they find is that there's if they measure electrical activity over the surface of the brain with a recording strip, that there's a gamma oscillation that occurs in concert with the hyperkinesias. And so what they have done is connected the recording strip to the stimulator, and when there's a sign of this gamma peak in the electrical activity of the cortex, turn down stimulation to the deep brain. Kinesias from occurring, but the patient continues to have control of their bradykinesia. The first example, or an early example of a closed loop stimulator where the stimulation that's occurring in the brain is driven by activity that's measured in another part of the brain. So this is really a concept that over time one can see how this could be used in multiple different venues. This example, I'm going to show a video. The video is actually showing here. Okay, great. So this is a video showing a very similar idea in someone with essential tremor, where there's a deep brain stimulator in the thalamus. Now, essential tremor only occurs when the person begins to move. So what they've done here is they have a second recording electrode over the surface of the brain that detects the signal that occurs before someone actually moves. And they use the connection to stimulate the deep brain when they pick up that activity. So the stimulator only stimulates when it's needed, when the person is about to move. Now, this has less in the way of preventing side effects, but one big advance is the stimulator is not going all the time, and it led to a tremendous increase in time before battery changes are necessary, which is important because battery changes here require neurosurgery. So another example of a closed loop stimulation. We also have stimulation experiments going on in people who've suffered stroke, with the idea that stimulating certain circuits, particularly in the cerebellum, may enhance their recovery after brain injury. There are also experiments going on in people that allow you to collect and monitor brain activity to try to correlate it with a particular behavior. And the video I'm going to show you now is from Dr. Edward Chang, who is an epilepsy surgeon and has patients who come into the hospital to try to find out where their seizures are coming from, where then neurosurgery can remove the locus of the seizures. But to do so, these patients need to have a grid of electrodes placed over the surface of the brain and record electrical activity for days to really hone in on where the seizure is coming from. Lucky and fortunate for the Brain Initiative, many of these patients volunteer to have studies done where their electrical activity is actually correlated with other behaviors besides seizures. And here, Dr. Chang is most interested in the ability to speak. So he will be collecting data over the surface of the brain that is responsible for the movements of the pharynx and larynx that give rise to speech, and has been able to actually decode this electrical activity to predict what the person is actually thinking about saying. And one can imagine for someone with amyotrophic lateral sclerosis, ALS, or a severe brainstem stroke where they can't talk, but they can still activate their motor cortex for speech, that this could be hooked up to a computer and they'd actually be allowed to speak again. So please show the video.
The proof that you are seeking is not available in books.
The proof that you are seeking is not available in books.
The proof that you are seeking is not available in books. Shipbuilding is a most fascinating process.
Shipbuilding is a most fascinating process.
Shipbuilding is a most fascinating process.
The next slide shows another example where instead of using electrical activity that's captured from underneath the skull at the time of epilepsy monitoring, a similar idea is using EEG activity that's captured over the outside of the skull by electrodes to actually help someone move a prosthetic device. So it may be that the information from inside can give us precision control over, say, language production, but we may be able to get from the more gross EEG signal the ability to enhance the control of, say, a prosthetic device. So this is done by Dr. Bin Hay when he was at Minnesota. The next video that I'll show you is an example of trying to bypass the anterior visual system in someone who's blind because their eyes are no longer functioning. But we can capture video information through, say, a video camera, and then try to transmit that information right to the back of the brain, which is the computational area for visual processing.
After losing his sight in a car accident, Jason Esterhazen never dreamed he'd see light or movement again. But with the flick of a switch,
wow,
his world suddenly grew brighter.
I still can't put it into words. I mean, from being able to see absolutely nothing, it's pitch black, to all of a sudden seeing little flickers of light move around.
While it's not normal sight, a groundbreaking device implanted in Jason's brain at UCLA Health allows him to navigate the world around him.
Being able to tell where a doorway is, being able to tell where the sidewalk begins or ends, or where the crosswalk is, are all extremely meaningful events that can help these people regain some form of independence.
Dr. Nader Poradian implanted the device made by Second Sight over the visual cortex in Jason's brain. The Orion device converts images from a tiny video camera on a pair of sunglasses into a series of electrical pulses. Those pulses stimulate electrodes in Jason's brain that let him see patterns of light that act as visual cues.
We basically have the video camera and the video processing unit functioning or performing the functions of what the eye normally does, and we go directly back to the brain.
By moving his head, Jason can distinguish light areas and objects from dark.
There's little white dots on a black background. It's like looking up at the stars at night.
Only the world's second person to receive the device, Jason is helping the technology to grow more useful as he learns what each flicker of light represents.
It's someone moving across the room, walking past me or walking away from me, or it's a light against the wall. It was just amazing to have some form of functional vision again.
Now he can perform everyday tasks he couldn't before, like sort laundry and safely cross the street. And with a little practice,
I'm closing the camera to show you that I'm not cheating.
Jason continues to expand what he can do. At UCLA Health, this is Barb Consiglio reporting.
Please show the video. So as you can see, there are many experiments going on now that could lead to short-term health benefits for patients with neurological, mental health, and substance abuse disorders. And these all involve manipulating circuits that are involved in those disorders. The other area where I think there's going to be a tremendous advance is in understanding the neuropathology that underlies different disorders. So currently, neuropathology is done under the microscope, identifying whether there's a loss of neurons, an increase in glia, a region of brain that looks damaged where the wires and connections between the brain areas are missing. But it's really quite gross, and it's not quantitative very much. The single cell transcriptomic analysis I talked to you earlier about, which is leading to the cell census project in, say, the mouse brain and soon to be the human brain, those technologies are already being applied to the analysis of diseased human brains. Say here, an example from Lee Yui Tsai looking at single-cell transcriptomic analysis in a small region of brain in people with Alzheimer's disease. It can actually see differences along the pathway of health to disease in Alzheimer's, and actually found really quite remarkable gender differences in the effects of Alzheimer's on the single-cell transcriptomics. The other example from Dr. Chung's lab at MIT using the Clarity technique enhanced to be able to see across large regions of brain, the brain cells and their connections. And this also can be done in postmortem human tissue. I should mention that the transcriptomic analysis that is right now revolutionizing neuropathology comes because it turns out that the RNA that's present in the nucleus of cells is almost as good at cell typing as the RNA that's in the cytoplasm in dissociated cells. And the importance there is that in autopsy brain, one cannot get the RNA from the cytoplasm from dissociated cells, but one can get the nuclei. And so these transcriptomic analyses in disease autopsy brain is occurring in the analysis of either RNA or chromatin structures in nuclei. And that was, again, something not really quite expected when we started, but has really transformed our ability to understand neuropathology. I'm going to pivot now to the discussion of neuroethics. And right from the beginning in the BRAIN Initiative, President Obama, he asked his Bioethics Commission to consider the ethical areas that are going to be challenges for the BRAIN Initiative. And they published 2 volumes, Gray Matter 1, Gray Matter 2. And then at NIH, we started a neuroethics working group that would help integrate neuroethics into the BRAIN Initiative at NIH. And this has been incredibly helpful over the time of the BRAIN Initiative. Now, what's special about the BRAIN Initiative in terms of neuroethics? Well, we talked about the 3 M's, which are the core principles in the BRAIN Initiative, to be able to monitor, map, and modulate brain activity and brain circuits. So monitoring brain activity in a human brings up some really tough questions, and particularly around privacy, particularly around what areas of thought one would capture activity for. We talked about activity that gives rise to speech, but could we think about activity that gives rise to your thoughts, your inclinations, your preferences? We talked about modulating brain activity in patients with Parkinson's disease or obsessive compulsive disorder, post-stroke. What about modulating brain activity to win a prize or learn math faster or do better on computer games. These are also issues that these technologies will actually challenge society to come to grips with. The Neuroethics group at NIH has put together a simple guiding principles for the BRAIN Initiative. Many of these are very intuitive, but really important in terms of helping the scientists, the human studies IRBs, to try and think through the consequences of some of this work. So safety being paramount, discussing with patients how the research will affect their capacity to take responsibility for their actions, autonomy, agency, privacy, for instance, predicting and planning for the malign use of these tools and technologies, which I hinted at a second ago, and advising caution in moving neuroscience tools and technologies into the medical and particularly the non-medical uses, to identify and address specific concerns of the public about the brain and how it works. The brain is really who we are, so it's really quite personal, and we have to be cognizant of how the public sees this work, understands it. And so to encourage education and dialogue with the public, and then to behave justly and to make sure that the benefits of this research is accessible to the people who really need it. So those are the kind of the guiding principles that the group put forward. An example of something that one should think about, Neuralink is a company. Elon Musk has had many successes in his career, moving science into the commercial space, and he and his company called Neuralink has developed breakthrough technology to record the electrical activity using a set of really fine but precise electrodes. This has been something that the BRAIN Initiative has been working on, and they have really exceeded all expectations. And this is going to be incredibly important in, say, folks who have paralysis and helping them to control a brain-computer interface, to move an arm or maybe walk again. But whether or not we want to use this technology to drive a car is something that society will need to think about over time. We are actually also funding research in neuroethics in areas around the BRAIN Initiative. So actual research in this space is ongoing. And some interesting papers have already come out, particularly with regard to the ethical issues around implanting patients with stimulators for research. What happens? What do we do? How do we take care of them once the research is over with, for instance? The BRAIN Initiative has been ably guided by two major reports. The first I mentioned, called BRAIN 2025, was the initial report that went to Dr. Collins, seen here in the middle. And it is kind of a Bible for the launch of the BRAIN Initiative. And many of the principles I talked about are outlined. I would encourage you to read that. It's available on the web, BRAIN 2025. There was a halfway point report as well, which was just delivered a couple of months ago to Dr. Collins. And here you can see Jim Eberwein, Catherine Delac, Francis Collins, John Mansell, Jeff Kahn. And these were the committee chairs for a scientific report and also a neuroethics report. So there's two reports that have recently been published, available on the web to guide the second half of the BRAIN Initiative. And in the second half, we intend to stay on the productive path that was already launched, supporting technology development and the targeted study of circuit components. In addition, there will be an emphasis on identifying not only the neural activity, but in precise characterization of the behaviors and quantitative analysis of behaviors, structures, and particularly in model organisms. We will balance individual investigator research, which generates the really groundbreaking ideas, with team science that allows you to get to knowledge that requires large-scale activity, such as understanding all the cells in the mouse brain, for instance, as a team project that can only be done as part of a team. And we are currently thinking about what would be the most impactful large-scale transformative projects. The mouse brain cell census project certainly would be a transformative project. What other type of projects will be the legacy for the BRAIN Initiative in the second half? And there are a number of which were suggested in the BRAIN 2.0 report to Dr. Collins at Tally. And we're very excited to have John Nye to come in to direct the BRAIN Initiative through its second phase. It's important to state that the BRAIN Initiative is not simply an NIH effort. There are multiple federal agencies involved, such as the FDA, DARPA, IARPA, the National Science Foundation for sure has been a tremendous partner in the BRAIN Initiative. There are also nonprofit partners, IEEE, the Simons Foundation, the Kavli Foundation, and these have all come together to form the BRAIN Initiative Alliance, which is a platform for funding opportunities and to promulgate the news and opportunities and achievements of the BRAIN Initiative to the public. The BRAIN Initiative is also a global effort, so there are very related BRAIN Initiative research programs going on in many countries, and they are coordinated, that they meet together at the International BRAIN Initiative to compare notes and to work, particularly in the neuroethics space, in a coordinated fashion. So there are BRAIN Initiative programs going on in Canada, China. The European Union launched the Human Brain Project just before we launched at NIH. There's an Australian Brain Alliance, Korean Brain Initiative, and the Brain Minds in Japan, with their relevant partner organizations. So just to close, I hope that I was able to convey the importance of filling in this gap in our knowledge about how the brain works, the excitement that comes when tools become available that allow people to answer questions that they thought never would be possible to answer during their lifetime. And that's not an understatement, because some of these tools have really been earth-shattering in their impact on understanding and seeing brain activity in action, in modulating brain activity, and indeed the ability to start to think creatively about how we can help people who have circuit disorders that cause them disability, whether it's chronic pain, opioid addiction, schizophrenia, depression, Parkinson's disease, and epilepsy, all the symptoms that a patient will realize are due to the disordered circuits that the pathology is causing. But it's really the circuit dysfunction that leads to the disability. So if we have, through the knowledge of the BRAIN Initiative, the opportunity to modulate those circuits for health, I think that the impact will be really everlasting. So stay tuned. You're going to hear from a tremendous cadre of investigators in the area of brain circuits and BRAIN Initiative, and enjoy and try and get hooked on trying to understand how the brain works. Thanks very much.
Thank you, Dr. Koroshetz, for that outstanding presentation. For additional informative presentations, view our agenda on our events page. And as a final reminder, our speaker will follow up with any questions you've submitted via email. Thank you again for your participation.